So today I'm going to talk about tonicity. So tonicity is going to be measuring the osmotic pressure gradients of two solutions that are going to be separated by a semi-permeable membrane. So you could just think about a cell and then its cell membrane separating it from the extracellular fluid or the outside of the cell. So to understand what tonicity is inside of the cells, you got to understand what osmosis is. And osmosis is the diffusion of water molecules across a semi-permeable membrane. So basically, the way that osmosis works is that water is just going to follow the solutes. So water is going to be moving from a low concentration of solutes to a high concentration of solutes. So I drew the three different types of solutions that we have. So the first one is going to be an isotonic solution. So in this isotonic solution, I drew here a red blood cell. It's anucleated, so it doesn't have a nucleus. So what you can see here, the little blue circles that are inside of the cell, these are going to be the solutes. And what I've drawn here is the water molecule, so that's H2O. And if you actually just count these molecules, in here you got one, two, three, four, five, and then on the outside in this extracellular fluid outside of the cell, you have one, two, three, four, five. And if you count the solutes, you got one, two, three, four, five, six, seven, eight, nine. So you got nine solutes inside and then nine solutes on the outside. So this is what a normal red blood cell would look like, or a normal cell in an isotonic solution. In an isotonic solution, it just basically means that there's no net water movement, or there's no osmosis of the water that's happening. Well, there is water crossing back and forth, but it's not one going higher inside or one higher on the outside of the cell. It's equal. Iso means the same. So what I drew here is basically the same thing. I just zoomed in on like the ICF, which would be the intracellular fluid, or the cytosol, which would be this inside portion of the cell. And then you can see the semi-permeable membrane, which would be the cell membrane. And then on the other side, you have this extracellular fluid, which would be the outside. So I just drew it like this so that you could see it as well. If you count the water molecules, you have one, two, three, four, five. Then you got the same amount on this side. And then you have nine solutes, and then the same amount on the other side. So that's just showing you that there's no net water movement, because the water would be following the solute. So that brings you to this next type of solution, where this would be a hypertonic solution. In the hypertonic solution, you can see that this cell is cremated, or it's shrinking. So tonicity is important because you need these cells to maintain their shape so that they could carry out their functions. So this cell has lost its shape, and more than likely it's lost its function. So again, this would be a red blood cell. And then here on the inside, you can see that there's three water molecules, and then that there's only one, two, three, four, five solutes on the inside. And because water is going to follow the solutes, so they're going to the higher concentration of solutes, which would be on this outside of the cell, which would be the extracellular fluid. So all the water molecules are actually leaving this cell, which would be causing it to shrink and lose its shape. So again, I drew the same thing. You can see the intracellular fluid, which would be this inside portion of this cell. You got three water molecules in here, and then only four solutes. So what's happening, pay attention to the arrows. You can see that the water molecules are actually traveling to the extracellular fluid. And then over here, I drew a bunch of solutes, so that's letting us know that there is a high concentration of solutes in the extracellular fluid compared to this intracellular fluid, where there's a low concentration of solutes. So these water molecules are actually they're diffusing across this semi-permeable membrane, which would be that cell membrane, to that higher concentration of them. So this would be a hypertonic solution, which would be causing the cells to shrink. And then if you look at the third and last solution, you have a hypotonic solution. And in the hypotonic solution, what you have is a higher concentration of solutes inside of the cell. So this red blood cell that I drew, it's supposed to be a lot larger or swollen compared to this first red blood cell that you see up here. And this is because there's a high concentration of solutes inside of the cell. So these water molecules that are in the extracellular fluid, they're diffusing across the membrane and actually going inside of the cell. And this causes the cell to swell, or it actually can lyse or burst. If it lyses, it's actually breaking open the cell membrane and all of its contents are flowing out. So I drew it again over here the same type of way, so you can see this semi-permeable membrane, which would be the cell membrane. Then you have the intracellular fluid, which is this inside portion of the cell, and you can see there's a high concentration of the solutes that are inside of the cell. So that's actually causing the water molecules to diffuse to the high concentration of solutes. That's all water is going to do through osmosis, is going to follow the solutes. And this is how tonicity affects our cells.